Hello, my name is Dariusz Kurs. I'm from the Bydgoszcz University of Science and Technology in Poland, and today I would like to tell you a few words about the use of non-conventional cryoprotectors in the preservation of biodiversity. Biodiversity is very important for the agriculture as a suitable pool of genes is necessary to create new cultivars. So the preservation of biodiversity is crucial not only for the environment but also for agricultural purposes and breeding programs. Unfortunately, in the past few decades, a very disturbing phenomenon of genetic erosion is being observed. And genetic erosion means that the biodiversity on Earth is decreasing. For example, the number of cultivated cultivars is also decreasing, and this is unfavorable. Therefore, we need to develop some preservation methods, protection methods of biodiversity. And among various techniques of biodiversity preservation, cryopreservation is considered to be the most successful long-term storage method. In this technology, plant samples are being stored in dewar flasks filled with liquid nitrogen at the temperature of minus 196 degrees. At such low temperature, the metabolism of the cells is arrested, and therefore they can be stored at this cryogenic stage for a very long time. Of course, maintaining the tissues at such low temperature is not an easy task, and the plant cells need to be prepared for the stress of storage at such low temperatures. And usually, in order to prepare the cells for this stress, we need to use different type of cryoprotectors, which means chemical agents that will protect the cell from the influence of the low temperature. And in our study, we focused on bleeding heart, which is an Asian plant species. It is a member of the Papaveraceae family. It is also known as Lamprocapnos spectabilis or bleeding heart due to the very nice ornamental flowers. This plant is used not only as a decorative plant, but it is also used in pharmacology and cosmetology because it is a source of many valuable metabolites that can be used, for example, in cancer treatment, as in the production of antidepressant drugs, but also in cosmetology, as it was observed that the extract obtained from this plant species prevents the aging of skin. And therefore, the aim of our study was to test the usefulness of natural plant extracts. We use different types of plant extracts and nanoparticles and the cryopreservation of plant genetic resources. So our study included two experiments. Uh, in general, we used here the encapsulation-vitrification cryopreservation technique. In this technique, this is a multi-step technique. First, the explants are precultured in vitro on a medium with an increased concentration of sucrose and abscisic acid. Then the shoot tips, the meristematic cells, are being isolated. They are encapsulated in sodium alginate to provide them some additional physical protection. Then the cells are dehydrated, and finally they can be stored in liquid nitrogen, and afterwards, whenever we need to place our samples, our explants on a recovery medium to restitute a complete plant. And in our study, we performed two experiments. In the first experiment, natural supplements were used, and this included coconut extract, rye extract, oat extract, and sesame extract, all at the concentration of 10% that were added into the preculture medium in order to check if they will have any cryoprotecting activity. In the second experiment, gold, silver, and zinc oxide nanoparticles were added either into the preculture medium or into the alginate bead at two concentrations 5 and 15 ppm. So, as for the results, we observed that the addition of plant extracts into the preculture medium did not improve the recovery potential of the studied two cultivars of bleeding heart. Some of the extracts even decreased the value of this parameter, for example, sesame extract in cultivar gold heart, but we observed that the addition of coconut extract into the preculture medium stimulated the proliferation of shoots during the recovery step. So we were able to recover a greater number of shoots. Although we did not observe any increase in the other of the studied parameters here, such as the number of leaves, the shoot length, or the shoot weight. On the other hand, nanoparticles were a little bit more effective, and their effect was also dependent on the nanoparticle type, the concentration, but also the cultivar and the moment of application, depending whether they were added into the preculture medium or to the alginate bead. And for example, we have noticed that the addition of silver nanoparticles and gold nanoparticles at low concentration into the recovery medium had a positive into the encapsulation bed had a positive impact on the recovery of the stored samples in liquid nitrogen. On the other hand, the addition of nanoparticles into the preculture medium had a negative impact on this parameter in comparison to the control where no nanoparticles were used. This was even more evident with the other cultivar, with gold heart. Here we observe that the addition of nanoparticles into the alginate bead matrix increased the recovery potential of the cryostored shoot tips by even 20 or 30%. And on the other hand, some of the nanoparticles added into the preculture medium had a negative impact on this parameter. Interestingly, the situation was a little bit different when considering the number and length of the produced shoots after storage in liquid nitrogen. Generally, we observed that the addition of nanoparticles either into the preculture medium or to the bead matrix did not increase the proliferation rate, the number of shoots produced after the storage in liquid nitrogen, but the shoots that were produced after storage in liquid nitrogen were significantly longer if nanoparticles were added into the preculture medium as compared to the shoots produced from the experimental objects in which the nanoparticles were added into the alginate bead matrix. And a very similar tendency was observed with the second cultivar. Also here we have noticed that the length, but also the number of shoots produced was smaller if the nanoparticles were added into the alginate bead matrix, whereas some of the combinations where gold or silver nanoparticles were added into the preculture medium increased the value of those parameters. So, to sum up, the supplementation of preculture medium with coconut extract stimulates the proliferation of shoots of bleeding heart after cryostorage. The effect of nanoparticles is strongly connected with their concentration and method of plant treatment, depending on the addition of nanoparticles to the culture medium or to the alginate bead matrix. Silver, gold, and zinc oxide nanoparticles applied at low concentration, 5 to 15 ppm, during the encapsulation step of the cryopreservation protocol have a beneficial impact on the survival of the samples stored in liquid nitrogen. And finally, future studies will focus on the utility of other nanocolloids such as copper or titanium in cryopreservation studies, particularly when added into the loading solution or to the plant vitrification solution, which are used during the dehydration step. We would also like to study a little bit more the effect of nanoparticles on the broadly understood stability of the plant cells in order to check if no changes, for example, at the genetic level have occurred. If you are interested more in our research, I invite you to read some of our manuscripts which are related to nanotechnology and cryopreservation. And if you are interested on working with us, we are always open to cooperate, so you are always welcome to contact us. And thank you very much for your kind attention.